Okay, so today we're going to talk about viruses. You may want an extra piece of paper to take notes on. It's probably not too much extra stuff, but just in case, it's up to you. Okay, this first slide you do not need to write down. These are just some viruses you may have heard of or come into contact with. Just going to kind of briefly talk about them. The influenza virus or the flu virus, probably something you've all caught at one point or another. It's actually not one virus. It's actually lots of different strains of the virus, including the H1N1, which was a big deal a couple years ago. The same is true for a cold virus. Cold viruses are rhinoviruses, and they are actually also many different strains. So every time you catch a cold, you actually catch a different cold. HIV, very well-known virus as well. SARS, it was a couple years ago, or probably a while ago now, when it caused quite a problem in the East Asian area, and that's a coronavirus. And a lot of you have probably either been vaccinated against chickenpox or have had chickenpox, and that is a varicella zoster virus. So there's lots of different types of viruses, and just keep in mind that what we're talking about today is very basic in terms of viruses. If you actually wanted to specialize in virology, which is the study of viruses, you would probably spend years and years just studying all the little details. But today I'm just going to give you the basics. One of the first important things to know about viruses is they are currently considered to be non-living. And the reason that they're considered to be non-living is because of two things. Number one, they don't have cellular structure. They don't have a cell membrane. They don't have ribosomes and organelles and things like that. They do have certain aspects that living things have, but not cellular structure. And on top of that, they can't reproduce or replicate by themselves. They have to actually use another living cell to replicate or copy themselves. So they're really considered more infectious particles. Generally speaking, viruses are also really small. You can see this in the picture right here, which you also have in your notes. This little spider-like guy, this is a virus. Compare that to this ovally shaped thing. This is a bacteria. And in the back, you have the human cell, the animal cell. So you can see there's a big difference in size. So viruses are very small in size, even compared to like a bacteria, which is already really small themselves. So let's talk about the basic components that you're going to find in pretty much every virus. The first two you're going to find no matter what. The third one is the one that I guess you could say is not always there. Even though viruses are not living, they all have to have the instructions on how to make more viruses. So all viruses are going to have some form of nucleic acid, either DNA or RNA. And this is actually also where they break some of those rules, because some viruses are single-stranded DNA, some are double-stranded DNA, some are single-stranded RNA, some are double-stranded RNA. So either way, they're going to have some sort of nucleic acid, which is the instructions on how to make more viruses. Surrounding that nucleic acid is a protein coat that's called the capsid. And then the third component that some viruses have but not all is what we call the viral envelope. That's usually derived from the host cell membrane. So the idea is that when that virus is leaving the cell that it's just used to copy itself, it kind of pinches off a little bit of its host cell membranes. This is something you don't find in all viruses, so just keep that in mind, that the first two, nucleic acid and capsid, you're definitely going to find in all viruses, but you don't necessarily find a viral envelope in all viruses. And you have this sort of generalized picture of a virus here, so we can go ahead and label that. We've got our nucleic acid in the center, we've got our capsid outside of that, and then in some viruses, again, we're going to find the viral envelope. Take a look at that viral envelope for a second. I'm going to have you talk to your neighbor about it for a moment. I told you that some viruses have viral envelopes, so some don't. It is generally considered to be potentially an advantage, right? So I want you to think about, why would having a viral envelope maybe help a virus? think about what does it look like? Where did it come from? And why would that matter? So take a minute right now and talk to your neighbor about why might having a viral envelope actually help a virus. Well, if you look at that viral envelope, it looks like, and it actually came from a cell membrane, right? So some of the things you may have thought of were these. Number one, by having that membrane that looks a lot like the host cell, it might sort of actually serve as a way to help bind to the host cell. That's actually one of the things that scientists think having a viral envelope does for a virus, because it helps the cells to bind, the virus to bind as if it were another cell to its host cell. And also, since it now has that membrane, it looks kind of like one of your cells, or the host cells. So it's also thought that it may help protect or camouflage that virus from the immune system, at least make it a little harder for it to detect initially. It doesn't mean it won't be detected ever, but it may help with some sort of protection, camouflage, etc. The other thing that's important to know about viruses is that all viruses have a specific host range, meaning not every virus can infect every host. And sometimes it's not even a certain organism. Sometimes it's specific cells within that host. So just keep that in mind, that not every virus can infect everything, right? And just to give you a few examples, you don't need to write these down, but just to give you some examples, the rabies virus, for example, only infects warm-blooded animals. So your fish can't get rabies. Measles only infects humans. So your dog can't get measles. And remember I said that you actually can also have, sometimes it's not even just the organism, it's specific cells. So when you catch a flu and you feel like the cough and the phlegm in your throat and whatnot, and that's because the flu virus is actually specifically only able to infect tissues in your upper respiratory tract. It's not going to infect your toe virus, your toe cell. HIV is another good example. It only infects specific immune system cells. Either way, when we talk about any virus, we're going to kind of look at some basic steps of how the virus reproduces. Remember, it can't reproduce on its own. It has to get into a cell and basically take over that cell and use whatever's in that cell to make more copies of itself. So there's some general steps it always goes through. The first step is what we call attachment. That's where the virus actually binds to the host cell. And there's a variety in how that happens, so we're just going to talk about it in more general terms right now. Second, there's the entry step. That's where the virus actually enters the host cell. Sometimes the entire virus goes in, sometimes just the nucleic acid goes in. Now, sort of the main event, right? Once that virus gets into the host cell, now it's going to go through what we call biosynthesis. This is where that viral genome, the viral DNA and the protein are synthesized. Sorry, there's a typo in your PowerPoint here. Should be are synthesized. And we'll talk about that in more detail in the next few slides. Once you've put together all those little pieces of what's going to make more viruses, then you have the assembly step. That's where actually the new viruses are going to self-assemble into viruses, into the capsid, the DNA or RNA in the center, etc. And then lastly, there's the release step. And this is where those new viruses exit the cell. In some cases, it's going to kill the cell in the process. In some cases, not. But either way, now all those new viruses can now go out and infect more cells. So let's take a better look at this biosynthesis step, because I don't just want you to know that, oh, they reproduce and that's it. Because now you have a little more background on protein synthesis, you can actually also get a better idea of how exactly does it copy itself. So you have this diagram in your notes. I'm just going to walk you through the numbers. So in step 1, which you see up here, right, the virus is going to infect your host cell. And next what it needs to do is it's going to take over the host cell's RNA polymerase, and it's going to first transcribe a set of genes that we call the early genes. Essentially, these early genes then code for early proteins, and these early proteins are going to do a variety of things. If you go down here to number 3, you can see that one of the early proteins is going to actually stop the host cell from transcribing its own genes. So it not only takes it over and uses it to make copies of the virus, it's no longer going to produce its own proteins. And another gene, early gene, is going to stimulate the viral genome to now be copied. So it's going to shut down the cell's own transcription translation, and now it's going to promote replicating the DNA or RNA of the virus. The third early gene is going to produce an early protein that basically triggers the next step, which is our late gene transcription. And the late genes, when they get transcribed, they produce proteins that are going to lead to the production of your new viral capsids, and a protein is also going to help, in many cases, break open the host cell when those viruses leave. There's not much you really need to write down in this slide. I just wanted you to understand what's going on so that it's not just a very general comment about, copying viruses, that you understand sort of how it does that. Okay. All of that was really more general to, viruses in general. But we're going to now start to focus on our specific viruses. The first type of virus we're going to talk about is what we call a bacteriophage. So we humans are not the only ones who can get infected by viruses. In fact, bacteria can also be infected by viruses, and the viruses that infect bacteria are called bacteriophages, or phages for short. They have a really distinctive looking capsid. You can kind of see you've got the nucleic acid in the center here, and then the capsid forms this really interesting shape where it looks almost like this sci-fi spider-like thing. What's actually going to happen is these bacteriophages, they are the type of virus where they actually don't have the whole virus go into the host cell. They just inject their DNA into the host cell, and you'll see an animation of this later. So we're going to take a couple minutes right now to talk about the two different ways that bacteriophages can infect and affect a bacterial cell. So basically, I guess you could say we have two different types of bacteriophages, and they can go through what we call the Lytic cycle or the Lysogenic cycle. So keep in mind now what we're talking about now is just for the bacteriophages, not for every virus. When a bacteriophage goes through the lytic cycle, first of all, this is going to result in the death of the infected bacteria. If you have a bacteriophage that can't only carry out the lytic cycle, then we call them virulent phages. And the lytic cycle is sort of like the death and destruction cycle. The bacteriophage go in, they infect the bacteria, they copy themselves, and when they leave, they basically blow up the cell. So we're going to look at that in the next diagram. Now you have this diagram in your notes, and there isn't too much you need to add to it. Again, it is a cycle, so it kind of keeps happening over and over again. But you can say here we've got the attachment phase, so we're going to see those general phases of viral reproduction here. This is your bacteria right here. This big circular thing inside the bacteria is its DNA. So the phage DNA gets injected in. It actually will cause the viral DNA, the bacteria's DNA to be chopped up and degraded. Right? Then it's going to synthesize all the viral genomes and proteins, assemble them together, and then when it bursts out and releases those new viruses, it actually will kill the host cell. So this is the lytic cycle. It goes in, copies itself, bursts out, kills the cell, right? It's a death and destruction cycle. Some bacteria go through what we call the lytic cycle, but other bacteria go through what we call the lysogenic cycle. So this one, for this picture, what I'm going to do is I'm actually going to be looking at half of the picture at once and then the rest of the picture. But first let's talk about the lysogenic cycle, and then we'll talk about how you should view the picture, or at least what you'll see in these lectures. Now, before we do this, it sounds like the bacteria, are pretty, vulnerable then, right? So I want you to think back to a different lecture. Bacteria aren't totally helpless. They actually have a way to protect themselves from bacteriophages, which are foreign DNA. So take a minute and talk to your neighbor. Can you remember any way, any method that bacteria have for protecting themselves from foreign DNA? Take a minute, talk to your neighbor about that. About a week ago, you talked about restriction enzymes, right? Remember I told you restriction enzymes were there naturally in bacteria to chop up foreign DNA. So that's actually how some bacteria are able to protect themselves from bacteriophages. They actually have restriction enzymes that cut them up. All right, let's move on with the bacteriophages. A temperate phage is a bacteria that can either go through the lytic cycle or the lysogenic cycle. So it can actually go through either one, depending on the environmental conditions, which we'll talk about in a moment. Now in the lysogenic cycle, it's going to sound a lot milder because the phage doesn't cause the bacterial host to burst and die. But you can also think of it as the sneakier cycle, because this is almost more like, instead of death and destruction, this would be like if the bacteriophage is like an undercover spy that gets into an organization and infiltrates it and then is able to cause damage on a big scale later on. And I'll show you what I mean by that in a second. So in this picture, you'll notice I've intentionally covered up the left side of the diagram, and that's on purpose, just so we can focus on the lysogenic cycle right now, which is what you see on the right side of the diagram. Again, we've got this bacteriophage, right? It injects its DNA or RNA into the bacteria. And now the key is the lysogenic cycle, I told you, is more like an undercover spy. So the DNA from the bacteriophage basically goes into the lysogenic cycle. If that DNA right there, it actually sort of becomes part of the bacteria's own DNA. It integrates itself into the chromosome. So what that means now is every time the bacteria copies itself just to reproduce, it's going to also reproduce the bacteriophage DNA. So it's secretly making a copy of bacteriophage DNA along with its own. So now instead of one infected cell, you've got two. And when they reproduce, now you've got 4, and then 8, and then 16, etc. So the lysogenic cycle, again, is just this half of the diagram. It can do this indefinitely for however long. There's no set amount of time or number of divisions. But remember, if we're talking about a temperate phage, it means it can go through the lysogenic cycle or the lytic cycle. So occasionally certain chemicals or radiation will cause a bacteriophage to switch from the lysogenic cycle, which is the undercover, copy itself covertly cycle, over to the lytic cycle. And here's where we're now going to uncover the other half of the diagram. Now, when we're in the lysogenic cycle, the phage is dormant, right? It's not really doing anything. It's not copying itself in any sort of large fashion. It's just copying its DNA with the rest of the bacterial DNA. Now here, when we uncover the rest of the diagram, here's where you see it switch to the lytic cycle. Again, when it says certain factors determine, those certain factors are certain chemicals and radiation. So what will happen is now the viral DNA will come out of the chromosome. Now it switches to the lytic cycle, which we've already talked about. It basically starts copying and making lots of copies of the virus, assembling them, bursts out, and kills the cell in the process. Okay, now's a good time for you to take a little stretch break if you haven't yet. Okay, we talked about viruses that infect bacteria. Now we're going to talk about viruses that infect us, animals, and other animals. So there's some really distinctive things about animal and human viruses. And the first one is that a lot of them do have a viral envelope, that third sort of optional component. And there are glycoproteins, so it's a combination of protein and carbohydrates that are attached to the viral envelope. And those are important because they actually help the virus to bind to your host cell. A lot of times people will say, okay, why would a virus have stuff, like why would we want to help the virus bind to our host cells? Not that we don't want to do that. Remember but think about where did that viral envelope come from? The viral envelope came from your cell itself, your host cell. So it's sort of like it's just taking advantage of the fact that it pinched off some of your own cells, and now it can use that to also get into another host cell. So this picture you have in your notes, and it shows you now a little more specifically the human or animal viral infection pathway. Here we have, in this case, a animal virus. It has the nucleic acid, the capsid, and the viral envelope with glycoproteins. So in step 1 for attachment, what happens is those glycoproteins on the viral envelope bind to receptors on the host cell. This would normally be how the cell is maybe using, other cells or communicating with other cells, but now it's just sort of been reused for the virus's purposes. In the second step, when the virus enters the cell, now usually the whole virus, not just the DNA or RNA, will go into the host cell. So that's usually through endocytosis. Sorry. Third, we have our biosynthesis of viral components. Kind of talked about the general idea of how that already works, so we're not going to do that in more detail here. Then you would put together all the different parts of your virus, so all your DNA or your RNA, your capsid, proteins, etc. And then one of the big differences here then is when the viruses leave their host cell, they're going to leave essentially by exocytosis. So they're going to bud out of the host cell, and by doing that they pinch off a little bit of their cell membrane, and that's how they get their viral envelope. Something else I want you to kind of make a note of here. In the drawing, it's actually a little misleading because one virus goes in, one comes out, kind of looks pretty harmless, right? But in actuality, what's happening is one virus goes in and then tens of thousands of viruses are coming out. So that's really how an infection kind of builds in an organism, in that you might only be infected by one copy of that virus, but over time, as they infect more and more cells, they're making lots and lots of copies of themselves. Right. One of the other special viruses, I guess you could say we talk about, or one category of viruses it's important to know about is RNA viruses. Remember I told you that the nucleic acid can either be DNA or RNA, so we're going to focus on those RNA viruses for a moment because they really have some unique properties. If a virus has RNA as its nucleic acid instead of DNA, first of all, we call them a retrovirus. And it gets a little bit more complicated than this, but let's just keep it simple. Retroviruses, they have RNA in their nucleic acid, right? And so when they want to copy themselves, that's a little bit of a puzzle because you have RNA and you need to make more copies of RNA, right? And normally cells don't really have an enzyme that makes RNA from RNA. So what the RNA viruses, retroviruses carry with them is they also have another enzyme that they take with them called reverse transcriptase. And the name of the enzyme kind of tells you what it does. It reverses transcription. So instead of making DNA or RNA from DNA, instead you actually take the RNA and you make a copy of DNA. Now some people say, why would you bother doing this? But once you have the viral DNA in your host cell, now that viral DNA can go through the regular process of transcription to make lots of copies of RNA, right? And then those RNAs can be used for your new viruses. So you create a DNA template from your RNA, and you use it to make lots of RNA. It's a much more efficient way of trying to copy your virus. HIV is a good example of a retrovirus. And as we'll talk about much later in the year, when we talk about the immune system, it's also one of the things that has made it a little bit trickier in terms of finding a vaccine against HIV, because reverse transcriptase is not the most accurate enzyme. So when it copies, it makes some mistakes, and those mistakes make it actually harder for us to find a vaccine against HIV because it mutates fairly quickly. But we'll talk about that later when we get to the immune system. These next few slides I don't really need you to write down. I'm just going to talk to you about them really quickly just so you have an idea of sort of from a news standpoint why were certain things a big deal a couple years ago. You may remember a couple years ago there was a lot of concern about H1N1, which was a new strain of the influenza virus that was first detected in April 2009. They were also calling it the swine flu. Again, you don't need to write any of this down. They originally called it the swine flu because when they looked at some of the genes in the virus, they thought they were similar to genes in the flu virus in pigs. But later on, they actually found it to be really different. What was really interesting about this virus, though, is they did see that it had genes from flu viruses found in pigs, in Asia, Europe, birds, and humans. So it's what we call a quadruple assortment. This picture kind of gives you an idea of how they think that may have happened. That essentially, if a pig gets infected with a variety of different viruses, like from a bird and a human and a pig, then what you end up with is that if a single cell gets infected by all these different viruses, when they are assembling themselves, what you're going to end up with potentially is a mixture of the DNA from those various viruses. So then you end up sort of with a mix and match, what they call reassortment virus. Now, the reason it was such a concern a couple of years ago is because it was a new strain that hadn't been seen before. So most people's immune systems didn't have immunity against the virus, and that would cause a lot more infections in the seasonal flu. Because basically, what health, officials and such ideally want is what they call community immunity or herd immunity, meaning if enough people in the population don't have already had the disease, then the chance of it becoming this big epidemic is much lower, right? And so if you now have a virus that no one's ever seen before, you're going to have a lot more infections. It's going to spread a lot faster. And with H1N1, it seemed to be spreading quickly among young people as well. So that's why they're really worried it was going to cause a lot more infections than the seasonal flu. Okay, so we have reached the end of our virus lecture. And so what you're also going to do now is you're going to watch an animation. The animation is going to show you, it's actually a combination of the lytic and lysogenic cycles, but for those of you who are visual learners, it'll allow you to kind of see how the lytic and lysogenic cycles work. And this animation is also on the website, so by all means do check it out as much as you need to. Okay.